Hey, welcome back, everyone. Mr. Taylor here for part two of our muscle physiology lecture. And just to recap a little bit, the last time that we had our lecture, we were talking about the different parts of muscle fibers inside of skeletal muscles. And we were looking at the different protein fibers that make up the sarcomere, which is the subunit of the myofibril. And if you remember, we talked about the thick filaments, which were made out of myosin, and the thick filaments are the part that actually do like the pulling. So that's the part that actually does like the movement of your muscle. And then we had the thin filaments, which were made of the protein actin. That's what all those little yellow dots that form a helix are at the bottom of this picture, and they're covered up by tropomyosin and troponin. So we talked all about those proteins. Today we're going to learn all about what those proteins actually do and how they make your muscles work. So here we go. So there's this little thing called the sliding filament mechanism, and this is how myosin and actin work together. So basically, like I was saying before, myosin is the protein that actually does the pulling. It's what pulls your muscles together and allows you to apply force and gives you strength. And so basically, if you look at myosin, all the little myosin heads kind of act like little oars, and they do like rowing of a boat. So think about like a skiff from like a crew, like for rowing, where all the oars pull together at the same time. All those little myosins that form that thick filament are all able to move. And so if we go back and look for a second, you can see how all those little myosin heads stick out. And if you can imagine all those being little oars and they're all kind of like rowing and pulling together, they're going to pull your muscle in tighter and make it shrink. And so when those myosin heads start pulling the filaments together, they cause an overlap, and they cause basically all the filaments to slide. So all the thin filaments slide over top of the myosin. And when this happens, if you remember back to our parts of the sarcomere, the I bands and the H zones get smaller and smaller. They shorten, and then eventually they totally disappear. So here's what this looks like. So if you look at this picture, at the very top, this is showing us a relaxed muscle. And so in this relaxed muscle, you can see very clearly that we have over here around the Z disc, we have this whole I band of light area. We have this A band where we have like dark region. We have the H zone in the middle where we just have the thick filaments. So the A band has thick and thin, the I band has just thin, and the H zone has just thick. And so what happens is we have this alternating I band, A band, I band, A band pattern going on inside the muscle. When the muscle starts to contract, what happens is that the I band starts to shrink because we increase the overlap. So the thin filaments are getting pulled in closer. So we have a smaller H zone here. So we have a thinner H zone. We have a smaller I band and the same size A band. So what's happening is the A bands are actually getting closer together and overlapping the I band a bit more. And so this is a partially contracted muscle where it's on its way to contraction. And finally, at the bottom, we have a fully contracted muscle. And you can see that all the thin filaments have been pulled to completely overlap the thick filaments. And now there's no H zone, there's no I band, nothing but A band. So the whole muscle looks dark. So if you looked at a microscope, if you looked at a skeletal muscle under the microscope, what you would see is if you see the pattern of dark and light bands, you see A bands and I bands, you're seeing a relaxed muscle. If you see where the A bands are bigger than the I bands, you're looking at a partially contracted muscle. And if you see nothing but A bands, you're seeing a completely contracted muscle. So we can actually tell microscopically what's going on inside the muscle. And so this is how your muscles work, kind of like an accordion. So you can think of the top as the accordion when it's completely open, and then as you squeeze it, eventually it'll get completely overlapped and become smaller, and then it can expand back out again. And so every time you move a muscle, this is exactly what's happening. That whole entire myofibril, each one made of many sarcomeres linked together, they're all going to contract microscopically, a very, very tiny amount. But all those microscopic contractions along those myofibrils is going to cause the entire muscle fiber to contract. A whole bunch of muscle fibers contracting is going to lead to the entire muscle contracting. And you can test this out for yourself. If you put your hand on your biceps and flex it, you can feel how your entire biceps muscle gets like shorter and harder and like kind of like thicker as you do this because you're actually overlapping these proteins. And so when you eat protein and you're trying to build up muscle mass, this is what you're doing. You're actually building all these guys. You're building actins, troponins, tropomyosins, you're building myosins in the thick filaments. And as you build more of these, your muscles get bigger. So you're actually adding more layers onto this. So you notice how like this little sarcomere has like one, two, three layers. This one has two. As you increase the size of your muscles, you actually increase the number of layers in your sarcomere. All right, so moving right along. Here's another picture showing you the same thing. This is a little bit more of a detailed picture, a little bit closer up, but you can see up in the top, this top picture is obviously a relaxed muscle because we see some empty space, and the bottom picture is the contracted muscle. There's less empty space. And you can see how the myosin here is actually hooked up from the thick filament onto the thin filament, forming what's called a cross bridge. So it's like bridging across. And this means this muscle is under tension. It's being pulled. It's giving a force out. If you look at this top muscle, because it's relaxed, there is no cross bridge, there's no connection. And if you notice, all the little tropomyosin molecules are covering up all those myosin binding sites on the actin, so there's no way for the muscle to contract. If you look down below, they're kind of pushed out of the way, and you can see actin and myosin hook up. So this is kind of like the overview of how our muscles work. So now we're going to start looking at kind of like more of like the nitty gritty details of all this. So what we started looking at was looking in here at how actin and myosin work together. We're going to look at this a little more in detail. But basically, we're going to look at how nerves, how neurons are able to send action potentials down to the muscle cells, how the neurotransmitters are sent across to turn on the muscle cells and excite them, how that gets sent down into the sarcomeres to cause them to form bonds and start to contract, and that's going to lead to our muscle cells starting to shorten and have a muscle contraction. So, when we look at contraction of muscles, there's basically two things that are absolutely necessary for your muscles to contract, and those are calcium and ATP. Now, we all know about ATP from biology, of course. ATP is our energy molecule. So, adenosine triphosphate is going to power the contraction of our muscles. Very, very important molecule. But calcium is also a very important molecule because calcium kind of acts as our trigger. Calcium is the molecule that, when released, is going to allow the muscle to start to contract. So, if you don't have ATP, you don't have any energy for the muscle to contract. But if you don't have calcium, you don't have a molecule that's there to signal that the contraction can begin. So when we look at calcium being like really important to this method, if you look at number 2, it says the sarcoplasmic reticulum constantly pumps in calcium under active transport. So if you remember, we said the sarcoplasmic reticulum's job is to store calcium. It is always pumping extra calcium ions into it under active transport. So it's constantly getting pumped full of calcium, and it goes against its concentration gradient. So the sarcoplasmic reticulum is ready to just like release tons of calcium as soon as your muscles are ready to contract. So when we get a nerve impulse from a neuron, so when a neuron sends a signal to a muscle cell telling it to contract, the thing that's going to happen is that the sarcoplasmic reticulum is going to release that calcium it's been storing. So the whole time that you're sitting here at rest and your muscles are just relaxing, you're just hanging on to all this extra calcium. It's being pumped under active transport into the sarcoplasmic reticulum, waiting for a nerve action potential to tell it to release the calcium. When calcium comes out, calcium is going to bind onto troponin. And if you remember, troponin and tropomyosin were those two molecules that covered up actin on the thin filament. As soon as calcium binds to troponin, it causes this thing called a conformational shape. Because troponin is a protein, some things can cause it to actually like kind of transform, and calcium is that thing. And so as soon as calcium binds onto troponin, it like goes through this conformational change, and it pulls tropomyosin and actually yanks tropomyosin out of the way, and it opens up all those myosin binding sites, and that allows your muscles to contract. And that's what this little animation is down here in this lower right corner. So we're looking at this little guy right here is a myosin head. That's a little oar that's going to do the movement. And as you can see, there's a little thing that flies in, it connects onto it. That's ATP. But if you look down here on the thin filament, we can see all these little yellow globes. Those are all of our actin molecules. The purple things are troponin, and the blue strand is tropomyosin. And as you can see, this little yellow, I think it's yellow, little sphere kind of flies in and hooks onto the purple thing. That's calcium. So calcium causes troponin to change and move out of the way, and it pulls tropomyosin with it. And as soon as it does that, it opens up these binding sites, allowing actin and myosin two get together. So I always think of this as kind of like dating. If you've ever been trying to you know, talk to someone, have a little alone time with that special somebody in your life, and they have, I don't know, their friends around or people around that kind of don't allow you to be alone. It's kind of annoying. You can't have that special time. And so I kind of think of calcium as being like the ultimate wingman. And so if you think about it, like if you have, a good wingman when you're dating, they'll help you out a little bit and help you get like some alone time with that special somebody. And that's what calcium does. When it comes in, it is myosin's wingman and allows myosin and actin to, kind of hook up a little bit. And it does that by getting troponin and tropomyosin out of the way. So it's kind of like our dating lives, I guess. This diagram is just showing you kind of everything all put together. So what we see here is, number one, here's our nerve sending its nerve impulse, releasing a neurotransmitter. And this is stimulating. That's what this little red arrow is. It's sending a little excitatory process around the muscle cell down to the sarcoplasmic reticulum. And when that happens, so when that nerve impulse arrives, the sarcoplasmic reticulum all of a sudden releases all these little calcium ions. That's what these little purple spheres are. They come down and bind onto troponin and move tropomyosin out of the way and allow actin and myosin to hook up and contract the muscle. And as long as that calcium is present, this is going to happen. So the contraction cycle is a little more detailed part of that. And this is how we actually get the contraction between myosin and actin. So when we're looking at actin and myosin binding together, the first thing that we have happen is that ATP has to stick onto the myosin and give it energy, has to energize it. So when ATP hooks on to myosin, it breaks down into ADP, or adenosine diphosphate, and phosphate. So it breaks down into two parts and gives energy to the myosin so it's ready to start. Myosin then is able to hook up and attach to actin and forms this thing called a cross bridge because it bridges between the thick and the thin filaments. The next thing it does is this thing called a power stroke. And the power stroke is where it actually does like its rowing, it does its pulling. And when this happens, the ADP breaks off, allowing another ATP to hook up later, and allows the myosin to push the actin. And then the fourth step is when ATP attaches onto myosin, it breaks the bond and detaches it from actin. And you can actually see that here. So when we look at this diagram, you'll notice that the myosin head has a couple little things attaching onto it. One of those things is ATP, which is this little green sphere at the front of the myosin. And so if you notice, that green sphere hooks up, and then a little piece breaks off, and then another piece breaks off and flies away. So what we're seeing here is that the ATP is breaking down into ADP and phosphate. The ADP and phosphate detach, and then a new ATP is able to fly in and attach onto the myosin head. Here's what it looks like in a diagram. And so we can see in this diagram, when our calcium comes in and it's hooked up to troponin and tropomyosin, we open up those binding sites and myosin is able to form its cross bridge. It's able to hook up with actin. Notice it's got ADP and phosphate hooked up to it because it's already been energized. Now we see this power stroke where the myosin actually flexes, it bends, and it pulls the thin filament to the left in this diagram. And when it does that, the ADP and phosphate detach. The third step we see here is that ATP is going to attach onto this myosin head and break that cross bridge. So notice it disconnects from the actin molecule, and then it resets. So I think of it kind of like, if you think about like shooting a bow and arrow, in order to shoot the next arrow, you have to put energy into pulling the string back, right? And then once you pull the string back on the bow, now you're ready to fire another arrow. Or you can think of this like an old school, like think Wild West, like revolver. They had to like pull the hammer back on the back of the revolver in order to fire off a bullet. So they put energy in order to fire it. The same thing happens here. So we actually put energy in to break the connection between actin and myosin and reset myosin back into its original position. So notice it's pushed all the way back over to the right, ready to swing back to the left again. So it's kind of like winding a spring, you could think of it maybe. Anyway, so this cycle repeats. So after myosin gets reset, it forms another cross bridge, it does its power stroke, it gets re-energized, and then it goes back to its original position and continues on and on and on forever. Here's another diagram showing you the same thing about how actin and myosin are going to hook up and form a cross bridge, and then we're going to release the ADP, have the power stroke, and then ATP is going to come in and break the bond. So it's kind of a weird situation because we have ATP coming in last. So normally we think about like giving energy first, but it's actually the last step is where the ATP is attached to reset the myosin back to start another round. And so what's interesting about this is it leads to a couple of little issues. For example, there's this little thing called rigor mortis that you may have heard about. And rigor mortis is when after death, a body becomes really hard and rigid. The muscles become really tight. And you may have heard about like people that suddenly move, after death. And what this has to do with is that when people die, because there's no more ATP being produced, the sarcoplasmic reticulum starts to leak out calcium. It can't do its active transport anymore. And so calcium starts to leak out and it causes the muscle to contract. And this contraction will continue except we ran out of ATP. So we can go through step one where we're going to form this cross bridge. Then we can do our power stroke. And then, we can't get to step 4 because there's no ATP to break the cross bridge. So we get stuck in this step where the myosin is doing its power stroke. And so it causes the body to become really stiff and rigid and lasts about 24 hours. So if you've ever heard of rigor mortis before, that's where it comes from. A similar thing can happen with cramps. One of the ways that you can get cramps is if your muscles run out of ATP, all of a sudden the muscle can just become like really hard and stiff and feel like you just can't move it. And that cramp is because temporarily those muscle cells have run out of energy. And until they can get fresh energy brought into them, until they can start to make their own energy, they just get stuck. So if you ever wondered, why you get cramps, that's one way. All right, so that brings us to this thing called the neuromuscular junction. And the neuromuscular junction, or NMJ as we sometimes abbreviate it, is another important part of muscles that are allowing them to communicate with neurons. And so if we zoom out for a second and look at the outside of a muscle, all muscles are connected to neurons, and these neurons are called motor neurons. They're the neurons that actually turn on and control the muscles. And so if we look at a motor neuron, they're always connected to several muscle cells, and they form this thing called a motor unit. And so the idea here is that if you were to stimulate one motor neuron, it would turn on a certain number of muscle fibers. It might be 5, it might be 10, it might be 100. But every motor neuron controls a certain number of muscle fibers in its motor unit. Interesting little thing about these motor units is that they vary in size throughout your body. So in parts of your body that need the most strength, so think about like your legs for like lifting your body and running, your arms for picking up heavy things. The motor units are huge. So one motor neuron is going to turn on a ton of muscle fibers to allow you to have a great strength. But in other parts of your body, you don't want like great strength. You want very fine control. And in those parts of your body, you have really tiny motor units where you get closer to a 1-to-1 ratio where every motor neuron is controlling just a few muscle fibers. They're not particularly strong, but they're really good at finesse. And so if you think about it, where in your body do you need the most finesse? Mostly in your eyes for looking around, in your hands for manipulating things very delicately, probably in your tongue for like moving food around and producing speech. So those are the areas that we find have the highest ratio of motor neurons to muscle fibers. In the stronger parts of your body, you don't need that much finesse. You need a lot more strength, so you have these large motor units. Anyway, motor neurons put out these things called muscle action potentials. That's what they trigger inside of muscle fibers. And so basically, we're trying to create a situation where we're stimulating the muscle cell. And so where the motor neuron runs into the muscle fiber, we form this thing called a neuromuscular junction. And literally it's just called that because it's where the neurological part, the neuron, runs into the muscle and they form a junction. And so this is very similar to a synapse that we already studied. In fact, it's almost exactly the same as a synapse. We have an axon terminal on one end, which is the end of the neuron. We have a synaptic cleft in the middle, which is just like the little space between the neuron and the next cell. But instead of having another neuron at the end, we have the end of a muscle, which is called a motor end plate. So it's like a little specialized region of sarcolemma in between. And I'll show you a picture. This is what it looks like. So here's our motor neuron coming down and forming the neuromuscular junction with a muscle fiber. And if we look, we can see here's the axon terminal where the axon comes to an end. It gets kind of fat and stores the neurotransmitters. Here we can see the synaptic cleft, like the little break in between, and then we can see the motor end plate where we see the very end of the muscle cell. And this is where the receptors are. So very similar, almost exactly the same as a synapse that we already studied. So here's what happens inside of that neuromuscular junction. First of all, acetylcholine is the neurotransmitter used in all of your skeletal muscles. It's the only neurotransmitter used to stimulate skeletal muscles. And so we always see that in every one of these neuromuscular junctions. Acetylcholine is released by the neuron into the synaptic cleft where it's going to bind onto receptors on the motor end plate. And these are going to open up sodium ion channels. And if you think back to the nervous tissue chapter, you can remember that sodium was used to turn on our neurons, right? So when sodium went inside of the neuron, it caused them to become more positively charged and depolarize. The exact same thing happens here. So when sodium goes into our muscle cells, it's going to cause that muscle cell to depolarize as well. And so when sodium goes in, it forms this thing called a muscle action potential, which is the depolarization that spreads throughout the sarcolemma and it goes down the T tubules deep inside of the muscle cell and stimulates the muscle cell. So basically, that's what we're doing here. We're taking a nerve impulse from a neuron. It's releasing acetylcholine. Acetylcholine is causing sodium to go into the cell. The cell is becoming positively charged, and that positive charge is spreading throughout the muscle cell, causing it to get turned on. But we don't want to keep this going on forever. So we have this thing called acetylcholine esterase, which is an enzyme that breaks down acetylcholine so that if you want to move a muscle, think about how fast you can move a muscle and have it relax. It happens in like the blink of an eye, literally. So acetylcholine has to get broken down very quickly. So all around your muscles in that synaptic cleft, we have this acetylcholine esterase, which is just breaking it down almost as quickly as it's being produced. So it works something like this. When we look way up close here, here's acetylcholine in our little synaptic vesicles, and acetylcholine is going to be released into the synaptic cleft where it's going to bind to acetylcholine receptors on the motor end plate. And right out there inside of that synaptic cleft, we'll find this acetylcholine esterase breaking acetylcholine down so it doesn't stick to that receptor forever. And then it actually takes the parts that it breaks down and sends them back into the axon terminal where it gets reformed back into acetylcholine. So if you ever wondered like why you can move your muscles forever and not run out of acetylcholine, it's because you're constantly making it, releasing it, breaking it down, and just the cycle continues. Okay, so now we've looked at how the motor unit works. So we're looking at how a neuron sends a signal to the muscle fiber. We're seeing these acetylcholine molecules go into the receptors. The receptors are causing sodium to go in, and the sodium is running along the sarcolemma, running down the T tubules. And when it gets down to the end of the T tubules, it's actually going to cause the sarcoplasmic reticulum to release calcium. And if you remember, calcium was our wingman, it's our trigger molecule, which is then going to bind onto troponin. It's going to move troponin and tropomyosin out of the way, opening up our binding sites, allowing myosin and actin to combine together and form a cross bridge. This is going to lead to myosin bending and contracting, pulling the thin filaments together, causing the myofibrils to shorten and causing the whole muscle to contract. Wow. That's a lot, isn't it? It's kind of like a game of mousetrap. It's kind of like, you have to have, or like a Rube Goldberg machine, I guess, where you have somebody that, kicks a ball and the ball like goes down a ramp and then it hits into something else and it causes, scissors to cut a string or whatever. And that's almost what this is like. We have like 99 different steps that lead to the muscle contracting, but each one serves a purpose. Acetylcholine is telling the muscle that it's time to contract, the nerve signaling it. Sodium is carrying that signal down to the sarcoplasmic reticulum. The sarcoplasmic reticulum is releasing calcium, which is going to open up those binding sites. Actin and myosin, they want to hook up all the time. So as soon as they can, they hook up and start contracting. And that's what causes muscles to contract. So that brings us to one final thing we're going to talk about today, and that's relaxation. How do we make all this stop? And so when we want our muscles to stop contracting, as we know, acetylcholine is the trigger that turns the muscle on from the motor neuron. Once we get rid of acetylcholine, once it starts to break down from acetylcholinesterase, now that starts to end the process. So once we get rid of the acetylcholine, calcium now starts to get sucked back into the sarcoplasmic reticulum because it's no longer being stimulated to be released. So as calcium gets transported back to the sarcoplasmic reticulum, the trypomyosin and troponin start to cover up those myosin binding sites, and now actin and myosin can't hook up, and the whole muscle slides back to its normal size. So if you think about this, as quickly as you can, like, you know, flex a muscle, as soon as you can, blink an eye, this all happens. It's amazing. So just kind of think about it as like the process in reverse, I guess is the best way to think about it. This also brings us to this concept of what's known as muscle tone. And this is something that people talk about a lot, but I don't think a lot of people know what it means. Basically, muscle tone is how tensed your muscles stay. So if you feel your muscles, they shouldn't feel like raw meat. They should feel like they have some sort of like tension or like strength to them at all times. And the reason for this is that even when you're relaxed, even when you're not trying to contract a muscle, your brain is still sending signals through motor neurons to the muscle cells and telling them to stay a little bit contracted. So, in any given muscle, you might have like 2% or 5% of the muscle fibers that stay a little bit contracted all the time. And the reason why they do this is because the higher your muscle tone, the better your reactions are. So it's kind of like, your car. If you think about it, if you were like getting ready to go someplace and it was in the middle of the winter, you would want your car to be warmed up and ready to go. You wouldn't want to like, jump in and immediately floor it, as soon as you turn the car on. And that's what muscle tone is. It's basically keeping your muscles in a state of readiness. Um, Throughout the day, your muscles usually have some tone. If you work out a lot and your muscles are really strong, they can maintain a higher level of tone at all times. And that's why, like, the more fit you are, the higher your muscle tone, but also, like, the better your reactions are to things. On the other hand, there's this thing that happens. Your muscles, like when you go to sleep, they start to relax so that they can heal themselves and all that good stuff. And they become what is known as flaccid. The muscles become really soft and they almost have this like raw meat feel. They're just like super, they just don't have any sort of muscle tone at that point. This can actually happen very tragically to people that have nerve damage. So if somebody like has a nerve that gets cut or blocked somehow, the muscles just lose all their signals from the brain and they just become like completely soft, and it's really, really pretty gross. Uh, but anyway, that's a pretty rare thing that happens, except for when you sleep. All right, so just to review, here's our relaxation. So when we see our relaxation, it's kind of like the whole process of contraction in reverse. So our acetylcholine starts to go away. It's getting broken down by acetylcholinesterase. When it's going away, now we have no signal. We have no more sodium going through because we're not depolarizing the muscle cell. So now the sarcoplasmic reticulum, without getting a signal, starts to use active transport to suck up all of its calcium molecules. So it's using ATP. So even when your muscles relax, it's using ATP to absorb calcium back in the sarcoplasmic reticulum. As the calcium goes away, now troponin starts to change shape back to its original shape. The tropomyosin goes back over and covers all those myosin binding sites on actin. Now, actin and myosin, as much as they want to, can't hook up anymore. So the thick and thin filaments separate, the thin filaments slide back, the entire muscle goes back to its regular full length and relaxes, and that's it. And so all of this happens once again in just a few milliseconds. All right. Well, hey, that's it for today's lecture. Next time, we're going to do part 3, where we'll finish this up and we'll learn all about different types of muscle tissue and all that good stuff. So hopefully you guys learned a lot, and we'll see you next time.